Multidimensional scaling is a set of statistical techniques that are used to reduce the complexity of the dataset and visualize its hidden features. To better understand how it works, let's bring ourselves to the context of current evolution explorations. We know that evolution is stimulated by mutations, changes in DNA sequence. Assume there is gene X that scientists want to look at. In a cell, it's a long chain of 4 types of nucleotides. But for our purpose, let's use only the first letters of their names and store this information as a txt file. Take another organism with gene X. Its variant of gene X begins with AGG. Then it's possible to randomly draw a point at an imaginary space called sequence space. This point represents the first organism's gene X. If we add the second organism, whose gene is 1 letter different, we draw a point somewhere close to the first one. The more similar the sequences are, the closer the points represented in them are. The third organism's point, GGG, may be added as well, but following defined rules, it should be closer to organism 2 than to organism 1. Fill in the sequence space with more sequences corresponding to gene X. It should become clearer now why the method is called multidimensional. In a typical textbook question, there are 2 dimensions, time and speed, whereas biomolecules are typically very long and may have 1,000 potential places where the changes might take place. It would have been impossible to see this space except by looking at huge documents with some kind of numbers, distances between the points. And those don't even fit the screen size. So, MDS is used in order to reduce the dimensionality of data to 3D to enable scientists to look at meaningful representation of molecules, analyze topological features of sequence spaces, and draw conclusions about evolution. The beauty of MDS is that it helps to keep only significant characteristics of data and stay focused. Stay here for more.